along Christy Meiselbach. Welcome, my name is Christy Meiselbach and welcome to session 2 of the symposium speaking about consent and communication in genetic research. Appropriate communication with communities is essential for informed consent, community engagement, and culturally appropriate care in genomic research and clinical services. Reflections on the role of linguistic concepts and experiences in creating a shared language to enable the communication of complex scientific concepts will be shared with the intent of supporting broad application of the presented methods in other contexts. So we have 3 presenters today who will be delving into this topic. Our first presenter is Dr. Clement, sorry, I've got to get the biographies up. Our first presenter in session 2 is Dr. Clement Adebamowo. He directs the bioethics training program, which provides short, medium, and long duration training in research ethics in Nigeria. He is the principal investigator of the NIH-funded African Collaborative Centre for Microbiome and Genomic Research and the African Female Breast Cancer Epidemiology Projects, which conduct research on persistent high-risk HPV infection, cervical and breast cancer. He is also the principal investigator of the NIH-funded Indigenous project, conducting research into the use of Indigenous educational concepts to improve comprehension on informed consent in genomics and cancer research in Africa. So I think Angeline is going to start Dr. Clement's presentation now. Enjoy.
Hello. Today I will be talking about engaging Indigenous communities to improve comprehension of informed consent for genomics research. Addressing disparities in diseases and outcomes between high and low and middle income countries requires epidemiological and genomic research. In addition to persistence of infectious diseases and diseases of poverty, LMICs are faced with rapidly rising incidence of non-communicable diseases. The rate at which these non-communicable diseases are occurring and their co-occurrence with persistent epidemics of infectious diseases is unprecedented in human history and presents novel challenges. Addressing these challenges requires research to be conducted in the affected communities because the outcomes of research conducted in one community may not be readily translated into others, particularly where there is significant disparity in resource levels. Similar to high-income countries, there are levels of availability of resources and access to healthcare even within countries. Oftentimes, the highest prevalence of infectious diseases and NCDs, their risk factors, and the worst outcomes are seen among indigenous populations in those countries, both high and low middle income. Therefore, research and implementation science studies in these indigenous communities needs to be a priority. However, concern is expressed about the validity of informed consent for research when research is conducted in indigenous communities because of low levels of education and awareness of modern genomic and precision medicine. It is well established that for consent to be valid, participants must be given adequate information about the research that they've been invited to participate in. However, disclosure of adequate amounts of information is not sufficient. Participants must comprehend the information that has been provided to them. Comprehension is a process of simultaneously extracting and constructing meaning through interaction and involvement, typically with written language, but can also be with visual and spoken language. So comprehension has three main components: the material that is to be comprehended, the reader or the person that is expected to comprehend the material, and then the activity to which the comprehension is related, in this instance, genomic research. And several studies have examined the determinants of comprehension, and they have reported that the determinants include age of participants, their educational level, their reading ability, professional background, their cognitive status, and the level of oversight that's provided in the informed consent process. Based on these predictors, several interventions have been proposed to improve the comprehension of informed consent. We therefore decided to study the comprehension of informed consent among participants in a genomic study of breast cancer in Nigeria. We administered surveys on comprehension of informed consent to 106 patients and 109 controls engaged in a case control study of the genetic risk of breast cancer in Nigeria. We conducted audio-taped key informant interviews in a subsample of 8% of the participants. Most of the women involved in this study were married. More than half of them reported having received a high school education or higher. Most participants were able to read the consent form, and we did not find any difference in the ability to read the consent form between the cases and the controls. The time between our study and when participants gave consent for the genetic epidemiology study was longer for cases compared to control. Our findings were all the participants involved in the study reported that they were told the purpose of the study. And most of the breast cancer patients, 83%, compared to 53% of the control participants, reported correctly that the purpose of the study was to learn about the genetic inheritance of breast cancer. Most of the participants reported that they were told about the study benefits, and 68% of patients, compared to 47% of controls, reported being told about the risks of the study. In the qualitative section of our study, we elicited the following responses from some of the participants. A control participant said, the researchers want to see if cancer is an inherited disease or if something else is causing the disease. A case participant said the purpose of the study is to enlighten us more about breast cancer and ways by which we can prevent it. They are trying to find out if the gene in our body has cancer. Another participant said the purpose is that if one had already passed through the blood test that the researchers offered, it is possible to know if the disease is a hereditary one, so that measures can be taken to prevent it from continuing to spread. Yet another case participant said the researchers will be able to learn the cause of cancer in our body and to learn if it is hereditary, something we have inherited from our parents, and help us to know so we will not pass it on to our children. We concluded from this study that the skill of the research staff in eliciting informed consent was very crucial. We came to this conclusion because several other studies had reported low levels of comprehension of informed consent in genomic research in Africa. We surmise that the disease type may also contribute to the high level of comprehension that we found. Participants in breast cancer studies may be much more engaged because of perceived severity of the diagnosis. An educational level we found was a significant predictor of reporting being told the purpose of the study was a genetic epidemiology study. We therefore recommended that consent forms should be modified by substituting words such as in your genes with words like in your blood in areas where the term blood is used to signify genes, thereby focusing on brevity and clarity. We believe that simplified consent forms that use clear and linguistically relevant terms should be developed and tested with individuals participating in genetic research in culturally diverse settings globally. And we recommended that when the potential risks associated with a study are high, investigators might consider not enrolling participants unless adequate levels of comprehension can be assured. So with this background, we then moved on to design a new study, which we called Indigenous Linguistic and Cultural Concepts of Heritability and Comprehension of Genomics Research in Nigeria. The objective of this study was to explore linguistic and cultural concepts among indigenous populations who are participating in genomics of breast cancer research and use what we learn about these concepts to improve comprehension of informed consent. To identify these concepts, we conducted 12 focus group discussions with 10 participants each. We ensured balance within these groups by age, gender, and religion. We used guides that were carefully edited to avoid preloading with concepts that may inadvertently influence participants' answers. Each focus group discussion lasted an average of 1 hour, and they were conducted in the local language, Yoruba. All the discussions were recorded and transcribed. Further, we enrolled 25 individuals who are considered key opinion leaders and custodians of community oral traditions for key informant interviews. We used scenario-based interview guides that we updated based on the results of our FGDs. We carefully edited the KII guides to avoid preloading, and we employed reflexive methods during the interviews and noted participants' observations and subjective expressions. So this slide shows us at one of the venues of the focus group discussions in the participants. And here are female participants reflecting a mixture of people from different religious backgrounds and age. And here is one of our interviewers talking to a key informant. We used thematic approach to analyze the FGDs and KII. We reviewed the transcribed recordings and used an iterative process to generate an initial level of codes, which were evaluated for secondary and tertiary level codings. Then we grouped the related codes into families and created networks of hierarchical themes. We analyzed each network for emerging themes and kept memos containing contextual information during this process. We used the consolidated criteria for reporting qualitative research to describe our results. Our findings were that the respondents in general affirmed that the Yoruba believe traits like character, attributes like height, illnesses like sickle cell disease and psychosis were heritable. They affirmed that Yoruba have a culture of engaging private investigators and intermediaries to investigate family histories, particularly within the context of marriages. The respondents also reported that Yoruba have a lot of words, proverbs, aphorisms, idioms that indicate heritability, and that heritability is mediated as an essence in the blood rather than in the materiality of the blood. For example, ejerun literally means blood smells, but actually means the perception of heritability in the blood. This is used, for example, when a child is behaving in ways similar to other members of the child's family. Respondents also indicated that Yoruba are aware that some traits or illnesses are sex-linked and that they are passed down one sex in the family, or that some traits are recessive and may skip generations. Participants also expressed understanding of the concept of dominant traits by saying that is, one blood could be stronger than the other in the context of transmitting heritable characteristics. The concept of dominant traits in the mind of the participants was not gendered, so the stronger blood may be that of the male or the female. Based on our results, we created an online database of indigenous genomic words and concepts, and we expanded our target population to all sub-Saharan African populations and invited researchers to contribute to support the development of this database. The database is a robust and rich online repository of indigenous genomic words, concepts, and expressions. Presently, there are 5 indigenous languages represented, and these languages are spoken in 11 countries. The database website also shows our research methodology, and we encourage collaborators to replicate this methodology for similar studies in their population. We believe that this will facilitate communication and adequate comprehension of the consent process by African participants in genomic research. And this database will therefore enhance the conduct of genomic research in these populations. So this shows the invitation to contribute to the database. In conclusion, Yoruba are aware of concepts of heritability and have words, idioms, proverbs, aphorisms, and sayings in the native language that's used to effectively communicate these concepts, and these words can be appropriated for use in the context of genomic research. We developed an enhanced consent form that's based on our findings and uses these words and ideas from the native populations. And we're currently conducting a randomized clinical trial comparing this enhanced consent form with the standard consent form in an ongoing genomics of breast cancer research project, and we will shortly be reporting our results. Remains for me to thank my collaborators who worked on this project, interviewing participants, transcribing and analyzing data, and writing the studies. I've also listed here the funding sources and declared that the opinions expressed do not reflect the position of the funding organizations, and to thank the staff and faculty of the Center for Bioethics and Research in Ibadan, which was the base where this study was conducted. Thank you very much.
Amazing. Thank you so much for that presentation, Professor Adebamoa. It's amazing that you've been able to create such a database to, help bring, that language together and, help with that informed consent. Our next speaker is Dr. Gareth Banum. He is a clinical geneticist, genomic policy advisor, patient advocate, and clinician scientist. He leads a number of Aboriginal precision health projects. He's the director of the Academy of Child and Adolescent Health and a member of the Western Australia Ministerial Council for Precision Health. He's the founder of Project Y, Pilbara Faces, Cliniface, Life Languages, and The Suspense is Killing Me, which are initiatives to support precision public health. So thank you, Dr. Banum, and Angeline is now going to start his presentation.
Thank you so much for the pleasure to speak with you today. My name is Gareth Banum. I'm a clinical geneticist from Western Australia. I'm saddened that I can't be there physically as we share together and learn from each other, but equally it's fantastic to have the access to technology to share stories together. Today I'd like to talk to you about Life Languages, about a suite of different initiatives to learn together with Aboriginal communities. I'm talking to you today from the blue waters of the Swan River, where the Aboriginal narrative is that the Woggle, a great serpent, carved this river, the Swan River, as it passaged through from the coast into the hills. I come from the world's geographically largest public health jurisdiction, Western Australia. It's 25,000 square kilometres of territory, much of it made up of red sand, and a number of the initiatives you'll hear about today have been based, in fact, far from the coast and the rivers and amongst the communities living in that red sand. We're extraordinarily lucky to have decades of resources which combine data, clinical services and policy to translate things for community benefit in the areas of genetic and rare diseases. And you'll see here a couple of icons, the blue circular one and the turtle, just reinforcing the critical nature of the partnerships we have with the Aboriginal health care primary sector and within a health department where our Office of Population Health Genomics sits within our Public and Aboriginal Health Division. This combination of community partnerships and policy has been critical to guiding initiatives. Whilst those initiatives listed above have been going for at least 50 years, well, that's a tiny, small number in the time frame of, say, 60,000 years. I'm using that time frame as it is the length of the longest living continuous oral culture in the world, that is the Aboriginal culture. Language is so fundamental to everything we do. Without language, we cannot share stories. We cannot have meaningful exchanges. We can't understand what people's aspirations and hopes are, or very succinctly, language is culture. Language is also memory. Be that written in our various memory codes, for instance, our DNA language, our genotype, one of our life's languages, and also a code in which we can write information within. For instance, Wikipedia has been written into DNA strands. Also our phenotype, our oral and written stories that we share with each other, or we might share in a medical encounter with our physician. It in turn can be itself used to codify and turn into a memory language. And of course, then it's how these things interact with our environments, our places and spaces. Or in an Aboriginal narrative, songlines, and you can see in this artistic representation the depiction of a songline as it traverses the country of Australia. These songlines, these stories, are sung differently between men and women. They're sung, the layers of the stories are revealed differently as people age. The stories are wrapped around geological structures and astrological structures to enhance retention of their memory and provide a sense of connection and continuity. In some ways, you can think of this as analogous to DNA strands wrapped around histones to package the stories and maintain its integrity. Or to epigenetics. We spoke about how these stories are differentially told between males and females, how the various layers are revealed at different stages of a person's life course of their development. No, I'm just a clinician, and so I like to bring back everything to stories. In fact, of course, it's the encounters with our patients that underlie all this and that motivate this. And personally, in the realm of Indigenous health, this was particularly brought to my mind in 2006, seeing a family with a devastating combination of things, in which we did some genetic testing and found a variant of uncertain significance. And then the ultimate story to then try and actually figure out if this in fact was the cause, and the various layers of discussion and technological advances that needed to be embarked upon to try and clarify that. As part of this process, we were directed to our senior Aboriginal leaders group in WA Health to really begin discussions about genomics, about emerging technologies, about how this can and does transform health, and to be guided very much by them. Also to further promulgate the needs, for instance, in this case, for reference data to compare against, to make our testing equitable. And then, as mother is a necessity of invention and healthcare needs to be equitable and focused to those that most need. In fact, if you have the triple whammy of a rare condition, living in a remote area, and being Indigenous, then indeed the needs may be high, and therefore what better place to start with trying to address those needs, to start with implementing new technology such as back then in 2013 whole exome sequencing. Ultimately, after seeing these three children who were sequentially born and investigated over a period of 10 years, finding an underlying genetic variant that we thought was extremely likely to be the cause, but because of the absence of reference data, we could not provide certainty to actually know this for sure, and therefore to counsel the family appropriately, and also in this case unlock the possibility of drug repurposing, that is using an existing drug for a new indication without needing to go through years and more than $1 billion in drug development. Ultimately, by circling the globe and finding colleagues to help us be able to ultimately prove this case, was to transform this family's care. This then became instructive for us, and also a powerful story to work together with further Aboriginal families and communities. As the journey continued, we needed to build further reference data, in this case phenotypic reference data, and specifically 3D facial reference data. So we worked with Aboriginal leaders, healthcare providers in remote regions, with them very much as partners and leaders of the projects. We hosted the world's first conference looking at the combined themes of rare diseases and Indigenous genetics. We took research data that had been acquired and then worked with the community in a governance structure to implement that research data, in this case the allele frequency data, the genetic information, into our public health system laboratories and make that accessible for public health system use to immediately help transform medical care. We continued working with other regions in Australia, moving from the Western Desert to the Pilbara region to build upon our facial analysis partnership. One of the technologies from the facial analysis is face language, in this case converting a 3D facial image to a text-based description that's computer readable and standardized, that is human phenotype ontology terms. It continued with further programs working together with the community, and for instance with our undiagnosed disease program, a quarter of patients were Aboriginal and a quarter were from remote and regional regions. This was not because they had more rare undiagnosed diseases. It was in fact because their health journeys were much more complicated. So rather than being sorted out early in their diagnostic odyssey, they ended up in essentially what is an end-stage program. But serving the needs of these families has led on to other solutions. For instance, serving the needs of another Aboriginal family led to the establishment of an international data exchange mechanism between Japan and Australia. These combined needs and the tractable opportunities then led to an ongoing suite of initiatives to improve health service delivery and capacity building, in particular capacity building of youth as the future leaders and solution providers of their communities. One such youth-driven initiative is Life Languages, which in its essence is a universal Indigenous medical translator, or at least it aspires to be so, as launched as part of the United Nations International Year of Indigenous Languages in 2019. It's about retaining and empowering language, about creating more connected communities, about fusing ancient and old knowledge with latest technologies. It very much works through youth, who are variably medical students, allied health students, artists, language teachers, professional sports people, working together with their community members and their elders together to perform these translations and to guide how they are used thereafter. This is one of our inaugural Life Languages champions, Shamir Rind. Shamir's language is dying, and he's particularly keen to help retain that language because it fuses both his cultural and personal needs as well as his medical student studies and his interest in health. Like our other Life Languages champions, Shamir is a network machine, and like no other people can, these incredible youth, through their connections with their community, really can build these networks. Or this is Yalaloo Thomas, another Aboriginal medical student who last year was WA Young Australian of the Year. In this case, working together with his community, which is a 2.5-hour flight from our capital city and then about a 3-hour drive to a place here called The Block, working together with his community to perform these translations. Yalaloo also works in our 3D facial analysis initiatives and other precision health initiatives aimed at improving Indigenous health. Mechanistically, Life Languages takes medical terminology, combines it with layperson translations, and then with Indigenous translations of those terms, with a backbone of a computer-readable and standardized language, the human phenotype ontology. The human phenotype ontology is the lingua franca of precision medicine. So for instance, you can see here the term macrocephaly, big head, in Yangamara Aboriginal, Njantuwurtu, and then converted through to the respective HBO term, which can then be used, for instance, to be used with multi-omics analytics. But it's not just about the translations itself, it's also about the narratives, the stories, because language has a context. And so, for instance, here's a page from a booklet which Yalaloo created to explain DNA and genetic healthcare in concepts that resonate with Aboriginal narratives, and in this particular instance, again, with songlines and singers. It's about responding to community need, and so the same group of champions self-assembled early in the COVID epidemic to create language translations One page, plain language in language for the relevant communities, from concept to implementation in the communities in 48 hours. We continue to work together with the community with various capacity building and sharing projects. And ultimately, to combine the voice of the doctor, the voice of the people, the voice of the Indigenous peoples, to combine ancient wisdom with new technology, to deliver answers, to respect, to learn from and contribute to community, to build trust and empowerment. So what are some of the key learnings? Trust is everything. I've paraphrased here from Stephen Covey, a very famous business leader. Nothing is as fast as the speed as trust. Nothing is as fulfilling as the relationship of trust. Nothing is as inspiring, and you can read on. But really, trust is at the foundation of everything. As is care. When we think about data and the FAIR principles of findable, accessible, interoperable, and usable, we must also complement these with the care principles. How do we think about collective benefit and return of benefit to communities? How do we give communities the authority and the means to control? How do we partner responsibly with communities and ethically? When these things are addressed, we have the ability to deliver AI squared. That is a combination of ancient intelligence and artificial intelligence. Thank you.
Great, thank you, Dr. Gareth Banum, for that amazing presentation. There was so much amazing work in there that you were doing, and that your staff and youth are doing. Absolutely incredible. So our next presentation is from Associate Professor Donna Cormack and Miss Hannah Burgess. Professor Cormack is a researcher and teacher at the University of Auckland. Her research focuses on racism and its impacts on health, issues of data sovereignty and data justice, and transformative and anti-colonial approaches to research and teaching in Maori health. Ms. Hannah Burgess is a PhD student. Her research interests are around Maori futurisms, namely how through being in good relation to Puna and Mokopuna can navigate shifts in science and technology and envision future desires. She explores these Maori futurisms in her PhD research in genomics and her collaborative art practices alongside Maori creatives based in Tamaka. So enjoy the presentation that Angela is going to put on for you now.
Tēnā koutou, tēnei te mihi atu kia koutou, koe tai mai a Ipurangi, ki te whakarongo ki te kōrero nei. Ki ngā tangata whenua, huri no i te ao, tēnei te mihi. Ki ngā mati o te wā, haere atu rā. Huri no kia tātou te honga ora, tēnā no tātou katoa. Kia ora, I'm Donna Cormack, and my tribal affiliations are to Kaitahu and Kāti-Mamoi in the South Island of Aotearoa, New Zealand. I'm a teacher and researcher in Māori health, and I'm happy to be presenting with Hannah today, who I'm thinking alongside in the genomic space. Our talk explores how, as Māori, we can honour and uphold our intergenerational relationships through whakapapa, as genomic research and technologies become increasingly common in our society. Grounded in Māori worldviews, we consider the role genomic research and technologies have in realising our wider aspirations as Māori, namely to be in good relation. And we also consider how we can navigate genomics in a way that honours our whakapapa relationships within and between generations. We acknowledge that our ancestors have always found creative ways to engage with the changing world around us. And being grounded in Māori worldviews can continue to provide a way to think through engaging with science and technology. We can think through how we can be in good relation with new ideas and technologies, including those in the field of genomics. In this presentation, we're going to focus on population health genomics as a context. That is, genomics in relation to its application to public health and population health inequities, rather than in terms of specific genetic conditions at the level of the individual or family, or the genomics of tumours, viruses, or other organisms, or DNA technologies more broadly. And I'm now going to pass over to Hannah to introduce herself and speak.
Kia ora, ko Hannah tōku ingoa, hiri tēnei no Ngāpuhi Te Āti Haunui a Pāparangi. My name's Hannah and I am a descendant of Ngāpuhi in the north island of Aotearoa and of Te Āti Haunui a Pāparangi, which is the iwi of the Wanganui River. Yeah, I am a PhD student at the moment thinking in this space, and Donna is my supervisor slash colleague, friend, co-theorist, co-creator. I thought it would be useful to begin our kōrero to speak a bit into my journey as a PhD student in this space, and how returning to Māori ways of being, knowing and doing has allowed us to engage deeply, not only in genomics, but what it means to be well as Māori or as Indigenous people. I remember at the beginning of my PhD journey, I was finding it really difficult to get my head around genomics and what Māori engagement in genomics could look like. I vividly remember I had like this mind map I made where I had like genomics at the center and I had all of these arrows coming out of it. And it was like healthcare, research, ethics, legislation, tissues, blood and genes, data sovereignty, Māori worldviews, tikanga, colonization. I had just, it was a mess. It felt so complex and I wasn't seeing clearly. Like I couldn't make sense of this, not only for my PhD topic, but for my whānau and for my community. And it was then I kind of came to this realization, or was something that I'd already known, that as Māori, our world is relational and we exist through our intergenerational relationships. So surely this would be my starting point, right? So I'd realized that I'd been centering genomics like as it exists now, and then thinking about Māori worldviews and understandings as kind of like this thing that we can add on top of this existing space, or it was kind of like this, just this layer over the top of genomics. And it was so difficult because there is so much about genomics that is fundamentally misaligned with Māori worldviews. And so we need to think critically, not only about genomic science and research, but about settler colonial society and what it means to be well as Māori. So from there and from this shifting of the center, like from genomics to being Māori, it meant for us leaning really deeply into theory, into Indigenous theory. And it meant thinking around genomics, not just in terms of risk and benefits for the here and now, but beyond that, thinking beyond human populations and towards the impacts that genomics and the framing and narratives around genomics have on our Māori world. So now I'll talk through whakapapa and how this shapes our understanding of the world as Māori. Whakapapa is a concept that explains the origin and positioning of all things. Whakapapa derives from the root papa, meaning base or foundation. Whakapapa denotes a layering and adding to that foundation. So rooted in creation, generations layer upon each other, creating a reality of intergenerational relationships. Everything has a whakapapa, all phenomena, spiritual and physical, from celestial bodies, days and nights, through to the winds, lands, waters, and all that transpires throughout. Whakapapa is often translated to the Western concept of genealogy, which can confine it to the past and make it appear to be primarily focused on human relationships of biological descent. However, as Māori, we understand whakapapa as much more expansive. Whakapapa is just as concerned with future generations and how our past and future generations relate to the rest of existence. So in knowing something's whakapapa, or the layers that make it up, you can come to know how it came to be, how it relates to wider existence, and how it will come to be in the future. Whakapapa allows us to understand the world and all its complexities. It not only provides a body of knowledge, but a way of comprehending the universe, forming the foundations of Māori ways of being, knowing, and doing. And this establishes important guidelines for how to be in relation with the rest of existence. Through whakapapa, all of existence is whanaunga, existing in the natural state of whanaungatanga. Whanaunga can be understood as this notion of being in good relation. The notion of being in good relation within and between generations is a common thread among Indigenous peoples, weaving together the existence of Indigenous peoples, lands, and oceans worldwide. Intergenerational relationships through whakapapa are the central nature of all reality and are ontologically privileged in Māori thought. So for us, to be is to be in relation. We exist not as separate individuals, but as wider collectives that emerge from and are sustained by our intimate relationships with the natural world. From the land to the ocean and beyond, our descent is layered through our whānau, hapua, and iwi, our collective groups, to the whenua and the landscapes of these ancestral areas, including our mountains, rivers, lakes, and oceans. Within this worldview, nothing exists in isolation. In fact, the notion of isolation is a fallacy. Everything in existence is infinitely and complexly in relation all the time. There are no breaks, no stops, no ends. Life is a continuously unfolding whole. This shapes a reality of interdependency, where the well-being of the whole is dependent on the well-being of its closely related components, and vice versa. When these relationships are at their natural state of balance, we are well. So within this context, we can come to understand settler colonialism. Settler colonialism, in short, can be understood as the violent damaging and disrupting of our relational worlds, the damaging and disrupting of whanaungatanga. Upon damaging and disrupting whanaungatanga, new settler colonial ways of being, knowing, and doing have been established. Settler colonialism ontologically privileges isolation, reduction, separation, and severing, unnaturally placing the individual human, white, straight, cis, able-bodied Christian men, to be more specific, at the pinnacle of existence. On the basis of such ontologies, whanaungatanga is replaced by settler colonial relationships that are premised on systems of domination, exploitation, possession, and commodification. Genomics is not divorced from this context. Genomics is intimately intertwined with settler colonialism. So critical engagement with this is a necessary step of making sense of our engagement in this space in genomics. I'll hand over to Donna now, who will speak to this in more depth.
So, as Hannah has said, it's important for us to be attentive to the context within which discussions about genomics occur. And for Māori, this includes a context that's already and always racialised and colonial, and where, as Hannah's noted, prevailing approaches to science embody extractive and consumptive logics. In this respect, the work of decolonial theorists and scholars remind us how divisions between human groups are central to colonialism, that race became the line that demarcated colonizers from colonized, and that social relations and experiences, including genomics, all happen within a context that's already racialized. And so we need to always be attentive to the qualitative difference of lived realities above and below this primary dividing line, what Ramon Grosfoguel refers to as the transversal line. How are genomic technologies experienced in these different spaces above and below the line of the human? Māori know at a visceral level the practices of naming, labelling, categorising, measuring and surveilling that have been part of historical practices of colonisation remain part of ongoing coloniality. Māori scholar Cheryl Smith refers to this as cultures of collecting, whereby Indigenous peoples become objects to collect, to gaze at, to exhibit, commodified as tradable products, or consumed as exotic curiosities. Colonial systems produce new classifications to define and describe Māori as a population. State systems of defining who was Māori reflected worldviews that conceptualized identity in racialized biological terms that sought to break Māori down into parts, using narratives of blood quantum and proportions of descent, in contrast to the deep, interconnected, and multiple ways of relating and identifying for Māori. And in this sense, we can think about narratives that seek to define being Māori populations only or primarily through DNA as a continuation of these reductionist biological notions of identity that emphasize the parts rather than the whole, and that elevate biological conceptualizations over relational ways of being and thinking. From a whakapapa orientation, an understanding of genomics gives us information about one layer of our expansive existence. It's important because it's linked to us and thereby linked to our whānau. However, it's not understood as the core or the foundation of our existence. A whakapapa or relational perspective of genomics disrupts what is reproduced in prevailing approaches to population genomics by challenging the notion of a hierarchy of knowledge where, to use Sylvia Winter's terminology, biocentric notions of the human are at the centre, and by which westernized science has come to assume or presume a greater credibility than Indigenous ways of knowing ourselves. A whakapapa approach to genomics also challenges central assumptions of settler colonial science, including ideas of objectivity, reductionism, and universalism, and narratives of progress or development. Genomics can tell us part of our biological story. For example, it can give us information about how we connect to others in our immediate biological family. Genomics can also help us in understanding particular health conditions to guide diagnosis and decision-making around what people might want to do. And this research can be critical for families with certain health conditions. However, genomics is also employed to attempt to tell stories about Indigenous peoples that it can't or shouldn't tell. Many of the recirculating genomics discourses back up rather than unsettle broader colonial narratives. Genomics cannot tell us who we are. While genomics may provide some information about some biological links to some ancestors, this will always be a partial story rather than a full representation of the multiple connections and relationships that we have. In this sense, genomics cannot tell us if we are Māori or not. If being Māori is about having whakapapa connections, being descended from certain ancestors, then our genes are not a measure of that. In much genomic health research, however, population groups are used in ways that, as constructs in ways that are decontextualized and ahistorical. As Kim Tallbear reminds us in talking about the idea of Native American DNA, that Native American DNA could not have emerged as an object of scientific research and genealogical desire until individuals and groups emerged as Native American in the course of colonial history, end quote. Much genomic research does not engage with the discussions and debates that have happened in other spaces around what these population categories represent. Scientists tend not to situate their practice within the history of their disciplines and what Teferi Zabuki has critiqued as the white logics that are embedded in many of the statistical techniques and methods used in population genomics. We suggest that there's an active ignorance of the ways in which population groups are socially, politically, and culturally constituted. That means that genomic scientists and researchers do not engage with the critiques of race as biology, with the scholarship and theorizing from other spaces about the situated and socially constructed nature of race, including critiques from Indigenous studies and Indigenous researchers. It's entirely possible, and not that uncommon, for research to be published making population genetic claims about Māori as an ethnic group that pays no critical attention to the way in which the population is conceptualized, nor what the use of racial or ethnic categories might mean more broadly. In line with progress narratives that are foundational in colonial discourses, population genomics is also represented as containing both explanations and solutions for the racial health inequities created by settler colonialism. And in doing this, there's a real danger that the fundamental underlying drivers of population health inequities, that is colonialism and its attendant systems of oppression, including capitalism and racism, are minimized or dismissed. Health inequities represent a racialized colonial capitalist system functioning as it should, rather than something anomalous that can be corrected within the same logics that created the problem. And I'll just hand back to Hannah to finish off.
Kia ora, Donna. So from this, in contextualizing genomics in its broader context, we can understand where genomics has come from and where it is going. We can understand its place in the colonial project, and we can begin to understand what that means for us as Māori, if we choose to engage or not in this space. Importantly, we need to center our worlds. We can put our well-being, our whānau, and our communities at the center of our frame. And from here, we can collectively envision what it means to be well. We can envision what it means to be in relation with the many layers of our existence, through our communities, to our lands and waters, and all that makes us who we are. And it is from here that we can query genomics. In putting our collective well-being at the center and the futures we envision, we query the role, if any, genomics has in our envisioned futures. And we certainly do not want to be involved in research and practice that keeps us oppressed and keeps us unwell, and further from these futures that we envision for ourselves and our descendants. It is through whakapapa and whanaungatanga that we consider rather how we can be included in genomics, but how genomics can be included in our world and under what conditions. We consider what sort of agreements, relationships, and levels of trust are necessary for us to be able to honor and uphold our intergenerational relationships. Because genomics can have a place in our futures, we just need to be clear of that place. We need to be clear about the stories it can tell and the stories it can't tell, and what this is all doing in the way of our broader relationships. Genomics doesn't have all the answers, but whakapapa does. The creation, maintenance, and strengthening of relationships is the basis of our worlds, and also the basis for negotiating permissions and restrictions, as evident in many of our ways of being and doing and knowing as Māori. Our tupuna have left us foundations through which we can navigate genomics. And as the multiplicity of relationships, environments, and contexts that make up our world change through time, so too do our processes of navigating permissions and restrictions. This is something we've always done. Our knowledge systems have always been fluid and contextual. It is about connecting to these knowledge systems and envisioning what these can look like for us in our futures. Importantly, as Indigenous peoples, we need the time and space to be still and to sit in these complexities together, according to our own tikanga. We need time and space where genomics and the goals, ambitions of careers of non-Indigenous scientists are not at the center of our discussions. There is no one answer to how we might engage with genomics, and we don't have the answer today. But we know that it begins with a returning to Indigenous knowledge systems, a returning to who we are, Māori. Mōri ora.
All right, thank you so much to all our amazing speakers. Those presentations were absolutely wonderful and gave us a really good insight into, communication and informed consent. So now we're going to open up the floor to some questions and discussion with the presenters. So I guess I'd actually like to start off the discussion, and I have a question for Professor Adebamwo, and I was just wondering if you could comment on, I guess, the transferability of the Indigene study methodology in other, I guess, contexts or Indigenous communities, not just the African community, so say like in Indigenous populations or other populations like globally.
Thank you. That's a great question. So in anticipation of questions like that, we actually open source the methodology. So if you go to the Indigene Study website, we have all the protocols and we encourage people to use them so that they can replicate the study in whatever environment they are. It's when a lot more people use those methods that we will know what their limitations are. But it does appear to us like it's a universally applicable approach to kind of probe individual languages, local languages, for how they've always thought about heritability. Because I doubt that there's any culture on earth that does not think about these issues, about relationships and, how people resemble each other. Some of the ideas apply to animal husbandry, to farming. People know these things, but when we as scientists come to those communities and presume that, we know better, you should call it gene, you should call it genomics, then it becomes quite problematic in my view.
Thank you for that. And so I will open up the floor to any other questions from any other attendees.
Just to build on Clement's comments, he's of course right about the inherent knowledge in communities and cultures about concepts we phrase as genetics, I guess. The use of the language around blood and changes in blood is certainly something that is used locally. And there's a lovely video from the National Centre of Indigenous Genomics from about 5 years ago, using those concepts and those narratives to explain their genomics research. And Clement, if you haven't seen that video, it's worth a look. None of it will be news to you at all, but it is a lovely video.
Perfect. Do we have any other questions anywhere?
I don't have a question, but it's more a comment. First of all, the presentations were absolutely fantastic. It was so interesting. And I saw on the first slide of Donna and Hannah's presentation, deep excitement in today's scientific world, which I thought was just so, such an incredible quote, really. And I particularly enjoyed these presentations because thinking about, there's a lot of talk around health literacy and the idea that we need to teach populations and need to teach individuals about scientific concepts. And I feel like these presentations have really turned the perspective around to some extent to think about how genomics fits into existing understandings and worldviews and ways of thinking and communicating. And I find it really exciting, this idea of bringing genomics closer to individuals, families, and communities and the way that we live.
Perfect. So I do have a couple of questions that have come up in the Q&A section. So I've just got one comment from Jeffrey Craig. who has said, "Thank you all for your powerful, insightful and passionate presentations. We have a lot to learn from Indigenous viewpoints." So thank you for that comment, Jeffrey. We've also got a question from Claire Zamet. This is to Donna and Hannah. First of all, she would like to say amazing presentation. And she was just wondering, could you please talk about any initiatives in place to support Maori in having more direct control over genomic research and any tribal governance structures for genomic data, biospecimens that may have been collected in the past that affects them?
Kia ora, Claire, thank you for the question. There is a lot of activity happening in this space at the moment, but in terms of sort of establishing tribal governance structures or tribally led biobanks, that's probably still in quite early phases. I know that a lot of communities are interested in the idea of having much more sovereignty or self-determination over engagement in genomic technologies. I'm not involved in any of the current initiatives, but there are some tribal health authorities and tribal authorities that are working directly with university researchers around genomics activities. And I think they're sort of in the stages of kind of negotiating what that looks like for each individual iwi. Yeah, so I don't want to speak too much for other groups. My sense more broadly is though that a lot of the control and the funding is definitely still going through universities and other kind of private public health provider partnerships, and less funding is directly going to Māori communities to do work in the way that they might want to do the work. So sometimes I wonder, and this is just a speculation, not based on any current sort of collaborations, but I wonder how much, how different this situation would look if there was much more investment directly in resourcing to Māori communities. Maybe some of these collaborations wouldn't exist in the way that they do now if self-determination was actually resourced properly.
Amazing. Thank you, Donna.
Sorry, can I add to that? Because I think this is just such a critical point. There is an absolute power imbalance in funding, and the funding mechanisms are not everywhere, but in some places are still very much colonial. And I think, unless and until that power balance is addressed at that funding and decision maker level, that there is an enormous challenge.
Yeah, definitely. Like, I definitely think there's such a big move to have more, Indigenous governance within research projects, but it needs to go further than that. It needs to go all the way up to those, that funding and those initiatives to try and get, the funding back into the communities and have a lot more of their involvement in all aspects of research, not just, that end user groups. So that's amazing. I actually have a question for you, Gareth. So I guess you mentioned in your presentation that there's an underrepresentation of Aboriginal and Torres Strait Islander people in reference genomic data. And I guess I just wanted to know if you could comment on if there are any Australian initiatives or strategies that are trying to address this, and are there any sort of institutional barriers that need to be addressed in order to improve that reference genomic data?
There are several, and so at a national level, the National Centre for Indigenous Genomics has been working on this from a community engagement and capacity building basis forward, partly as negotiating in a sort of a careful and respectful way around an existing set of resources that were housed at the Australian National University, and discussing with the communities about if and how these should be used. And relatedly, there are initiatives which will be published soon around genomic sequencing reference data, but not just allele frequency, really looking at the entirety of the genomic architecture, because the differences are so much richer than just differences in allele frequency. There have been a number of initiatives, for instance, through Telethon Kids Institute, both in Western Australia and the Northern Territory, to build our whole exome sequencing referencing data. That was the stuff that we translated a few years ago into our public health system use. And there are other initiatives going on, for instance, in Queensland around renal health. There are still a number of issues around connecting that data, if you like, to meaningful data about people and communities, and by that I mean phenotype. Because ultimately, genetic reference data is rate limited by its connection to phenotype, to stories, and so that's really a key missing piece. In terms of challenges, I think there are a number, and I think the other speakers today really surfaced so many of those. But those challenges are also enormous opportunities. And one of the ways I think that's been very powerful is to have discussions with communities when we're really focusing on things that we can deliver against health leads right now. So things like, for instance, inherited breast cancer or rare diseases, or things where we know there is immediate clinical utility to this information, and that way we can solve real world problems in a tangible way and build relationships and trust through that. I think the power imbalance at the decision making and funding level is still an enormous challenge, not just for genomic testing or genomic research. And I imagine, like so many other key health challenges over time, that we could think about HIV, for instance, it's when the community itself aggregates, empowers, argues for, in a strategic and united way, for things to be done appropriately, in the right timeframes and the right scale. That's when we'll get the most significant change.
Amazing. Thank you so much, Gareth. So I've just received a question, and this is to all of the speakers, from Jeffrey Craig. And so he would like to know, to what extent does the concept of epigenetics fit in with Indigenous viewpoints of a broad concept of health?
I've spoken too much. I wonder if Donna or Clement would like to.
I mean, I think that's a really big question. Yeah. I guess where I see maybe some resonance, and this is my, just speaking for me and my understanding of Indigenous health for Māori, is epigenetics feels a little bit closer to a relational understanding of the world, in the sense that it doesn't kind of try to separate individuals out from the rest of the relations that people have, and those relations are not just with other humans, but with more than human kin, so with the land and with the rivers and the waterways. But I guess, yeah, I'm it's still in the way that it's I'm still thinking through some of the kind of issues with the way in which epigenetics is applied, that actually don't really deeply unsettle those ideas of kind of the individual biological organism interacting with their environment, to get to a deeper understanding of how connected we all are. I don't know if that made any sense. I guess I think it aligns it aligns in some ways, but in the way it's applied in science, it actually tends to draw on some of the same methods that I would probably critique in terms of the sort of statistical assumptions and things underlying its application and research.
Thank you, Donna. Did any of the other speakers want to contribute anything to that question or discussion? No? Okay.
I'm going to speak too much again. I think about this a lot. And also to be clear, I'm not Indigenous, at least. I'm Anglo-Indian, but I think about how Indigenous worldviews resonate and can instruct with different scientific lines of thinking or constructs, and so from the epigenetics point of view, that the concept that you have some sort of understanding that is revealed in different ways, but over different times of your life, in different environmental contexts and differently between different genders. I think it resonates, at least for instance, with the Aboriginal narrative around songlines and then I also think about connection to animals and plants and other organisms. And so, for instance, one of the really powerful things in genomics in the moment that is looking for sharing across species. We do cross-species comparisons to figure out whether something's significant or not, and to navigate our way through the sort of the complexity of biology. There's an incredible initiative called the Monarch Initiative, for instance, which does exactly that. And so I think it's is looking for those points of joint learning, and where we can have a journey around those concepts of connectedness.
I mean, I think something I might add there is sometimes where there are areas of kind of resonance or alignment at one level, there's also a potential risk that of the sort of appropriation of Indigenous concepts. And here I'm speaking from some of the presentations and work I've seen happening in relation to Māori communities, where concepts like whakapapa, which are really deep and complex, kind of get used in genomics projects, but without the context or the deep history or the actual connection to that concept. So there's kind of, I can see it can be positive and negative in the sense of it can create areas where people can see resonance with worldviews within their community, but it also, I've seen lots of examples of it being kind of appropriated as a way to sort of, in a very simplistic way, to kind of look like the science is more aligned with Indigenous approaches than it actually probably is at any deep level.
And that happens, all the time, where, that kind of language and stuff is used, and it's sort of like this token, tick the box. We've included some Indigenous language. We've had a mild thought about it, but the actual, I guess, center of those concepts actually hasn't been addressed within the research. So I think that's such an important point that you've raised.
I think you've got to be really, really aware of that exact tendency, and because I think it's a real threat, it's a real problem and a real issue. And one of the things that concerns me is when I see someone try to almost tack on something Indigenous onto an existing initiative, and often done for really great motivations by lovely people wanting to do the right thing, and rather than taking it back and saying, like, to the community, what do you think about this? Is this an issue to you? What's the right way to go about this? What's the wrong way to go about? Should we even go here? and starting from those just really human discussions first, rather than there's something rolling on and we want to bolt a new carriage onto the train. And when should we even be building the tracks, or is it something else that we should be doing? So anyway, that's just, again, as a non-Indigenous person working in sort of genetics and phenotyping, there's a feelings that I'm expressing it, and I don't pretend that I'm Indigenous or representing Indigenous views. They're my, personal sets of experience, I guess.
Thank you. Okay, so I've gotten a question for Gareth from Philippa. Gareth, do you feel in your own practice that a lack of Indigenous reference genome and associated phenotype data has impacted your ability to diagnose patients in a way that has impacted their care? For example, variants of unknown significance in the BRCA1-2. What can be done to ensure equity for patients in the interim period before these references are available?
Yeah, absolutely. I mean, for me as a clinical geneticist, it's my daily work, and recurrently, this is an issue for us. And it's both a false positive issue and a false negative issue. So, with the lack of reference data, and both those are really harmful. So, because you can misattribute disease causation to a variant, for instance, and if you say that's a BRCA mutation, and you suddenly say it's pathogenic because you've never seen it before, and the data sets you've got available, and you kind of do some modeling that supports that it probably is, and you do some other things and it looks like it is, and you end up making some sort of decision to use that information. And then you might do a mastectomy. And then 2 years later, you go, actually, sorry, no, that we don't actually think that was disease causing. It wasn't pathogenic. We didn't have the reference data, et cetera, et cetera. And that happens. And there are examples of this in the literature, this happening with inherited cardiac disease in the United States, and there'll be examples everywhere. But you see it both, and as you said, you see it both in terms of false positives and false negatives. And you see it in diagnostic and therapeutic delay. So that first family, that family that I talked about from the Western Desert, we were pretty confident we'd found the underlying genetic cause when we did the exome sequencing, but there was zero reference data. And it was a novel condition, and if it were a Caucasian family, we actually would have called it at the time we found the variant and said, that's it, we're on to, this is the cause, we're not worrying about any other potential causes, and by the way, we can repurpose the drug to help with your family. We'll start you on rapamycin. So we then went through 2 years of extra laboratory work and to try and do functional studies and improve this, and etc. So yeah, it's a recurrent problem, and it's a global recurrent problem. So there's a whole range of things to improve that. What to do in the interim, or you can continue to argue for these things to be done in culturally safe and responsive ways, but also be really cautious. So be really cautious about what you make a clinical decision based on. So if you have an Indigenous person, I think you need to have an extra layer of caution about your interpretation of the disease causation, the pathogenicity or not of a variant. So, I think just having that extra layer for clinicians, for instance, just having that extra layer of okay, I just need to think around this a little bit further. And I think we need, so we're being safe in the way that we approach things. It's very easy to give someone a label, a diagnosis, a black and white report that says something. It's incredibly hard to retract that, because once it's in the community speak, or it's out there, or it's in a medical report, and it's in a set of files, or it's in an electronic health record, it's very hard to undo, despite people's best attempts. So I think we need to keep working together to resolve these issues, and I think in a clinical sense we need to be very judicious in how we make clinical decisions based on genetic data in Indigenous populations.
Thank you, Gareth. So we're coming up to the end of the session. So I think we have time for one more question. If anyone has anything else that they would like to ask any of our presenters, sort of last chance to do that now. No. Okay, well, I actually have a question for Clement. So in your presentation, you mainly spoke about comprehension and informed consent at an individual level. So I guess there are some ethical questions relating to genomic research, which are around how to develop models of consent that take into account the familial nature of genomics research. So I was wondering if your work touches on non-individualized models of obtaining informed consent.
Yes, thank you for that question. So we do look at other areas of consent, and more recently, we've been devoting a lot of attention to consenting issues in Indigenous populations related to data science. So, with the modern innovations in data science, where a lot of data related to our daily activities, our movements, our phone calls, our shopping habits, all of this data is just being scooped up. And we have to think about the parameters around consenting in that kind of environment. So people sign up, for example, you buy a new phone, but at the time that you're buying the phone, did you really consent that the GPS data on that phone can be, collected, and maybe there's a public health emergency, and that data becomes a variable in somebody's study. So we're looking at all of those different issues. But for this presentation, I just thought it would be nice to focus on the issues related to comprehension of consent and what we're doing to kind of dig into that in some depth.
Perfect. Thank you so much for that. Okay, well, I think we'll wrap up today's session, our second session for today. Thank you all so much for attending. Thank you to our amazing presenters, and thank you for an amazing discussion. It's just so incredible to see all the amazing work that you are all doing in the Indigenous space in different countries, and it's absolutely amazing that we can all come together like this to share learnings and our experiences to, better inform our research and, keep us on, the right path. So thank you all so much. It's been an absolute pleasure speaking with you all today. And I think, yeah, perfect. All right. Thank you so much.